Hello, my name is Joe Shomgabi. I welcome you to Coordination and Control. Questions 41 to 45. Describe the physiological events that lead to apical dominance, 2. Lateral bud growth and branching. Part B, compare short day and long day plants. Describe the physiological events that lead to apical dominance. Apical dominance is largely controlled by auxins. The gradient of auxin and gibberellins from the apical buds, they attract food and cytokinins. The food substances, for example sucrose, from the leaf and cytokinin and amino salts from the root apex, they are translocated past the dormant lateral buds, which have a higher concentration of the growth inhibitor like abscisic acid and low concentration of cytokinins, auxin, and gibberellins. To the shoot apex, lateral buds are inhibited and they become dominant, while the apical nodes become dominant. Lateral bud growth and branching. Lateral bud growth and branching is largely brought about by cessation of auxin production from the apical buds and raised cytokinin production in the lateral buds. Removal of the apical buds ceases any more auxins production from the apical buds. The lateral bud becomes active and produces own cytokinin, auxins, and gibberellins. The growth inhibitor, like abscisic acid, at the lateral bud is inactivated. The food substances, for example sucrose from the leaf and cytokinins and amino salts from the root apex, are translocated to the dormant lateral buds. Growth is stimulated in the later buds, allowing branching to occur. Part B. Compare short days and long day plants. We shall start with the similarities. Flowering in both plants is controlled by the photoperiod. Flowering in both plants is controlled by the phytochrome system. Flowering in both plants follows the activation of the dormant buds by foligen hormone. Let's now look at the differences. Short day plants, flowering is induced by dark periods longer than the critical length. Long day plants, flowering is induced by the dark periods shorter than the critical length. Differences still. Examples of short day plants include tobacco, soya beans, strawberries, and many others. Long day plant examples, we have the cabbages, spring wheat, spring barley, and many others. Short day plants, flowers on exposure to far red light. And long day plants, flowers on exposure to red light. In short day plants, phytochrome far red is slowly converted to phytochrome red by prolonged exposure to far red light. Long day plants, phytochrome red 660 nanometer, rapidly converted to phytochrome far red on exposure to red light. In short day plant, phytochrome red induces flowering. In long day plants, phytochrome far red induces flowering. In short day plants, phytochrome far red inhibits flowering. In long day plants, phytochrome red stimulates flowering. Number 42. Explain what is meant by the following: dark adaptation, light adaptation. Part B. Describe the changes in the eyeball that result in each of the following adaptations. Part C. Outline the mechanism that enables us to see over a wide range of light intensities. Part D. Radiologists, aircraft pilots, and others who need maximum visual sensitivity in dim light wear red glasses before entering the dim-lighted room. Explain why is this so? Explain what is meant by the following: dark adaptation, Dark adaptation is process by which the person is unable to see the objects in dim light. If a person enters a dim-lighted room, that is a dark room, from a bright-lighted area, he is blind for some time and he cannot see any object. After some time, his eyes gets adapted and he starts seeing the objects slowly. The maximum duration for dark adaptation is about 20 minutes. What is meant by light adaptation? Light adaptation is the process in which the eye gets adapted to increased illumination. When a person enters a bright lighted area from a dim lighted area, he feels discomfort due to the dazzling effect or bright effect. After some time, when the eyes become adapted to light, he sees the object around him without any discomfort. It is mere disappearance of dark adaptation. Maximum period for light adaptation is about 5% or 5 minutes, sorry. Describe the changes in the eyeball that result from the above adaptation. Causes of dark adaptation. Increased sensitivity of rods as a result of resynthesis of rhodopsin. Time required for dark adaptation is partly determined by the time for resynthesis of rhodopsin. In bright light, most of the pigment molecules are bleached, but in dim light, it requires some time for regeneration of certain amount of rhodopsin, which is necessary for optimal rod conduction. Dilation of the pupil. Dilation of the pupil during dark adaptation allows more and more light to enter the eye. Causes of light adaptation. Reduced sensitivity of the rods. During light adaptation, the sensitivity of rods decreases. It is due to the breakdown of rhodopsin. Constriction of the pupil reduces the quantity of light rays entering the eye. Part C. outlined a mechanism that enables humans to see over a wide range of light intensity. Pupil changes, duplex retinal, that is, both the rhodate cones as photoreceptor cells, dark adaptation, and pigment bleaching, light adaptation of the visual system and individual neurons. Part D. Radiologists, aircraft pilots, and others need maximal visual sensitivity in dim light. Wear red glass before entering dim lighted area. Explain why this is so. Because red light of the spectrum stimulates the rod slightly, while cones are allowed to function well. Thus, a person wearing red goggles can see well in bright lighted area and also can see an object or objects clearly as soon as he enters the dim lighted area. Part 3. How is the motor neuron suited for its functioning? Part B. With appropriate example, describe the various types of reflex arcs. Part C. Compare neural cell with other cells found in the mammalian body. Part A. How is the motor neuron suited for its functioning? The nucleus is relatively large to coordinate metabolic activities all over the large cytoplasm of the cell. There are very many rows of nisyl granules for massive synthesis of proteins. Axon is long to carry impulses over a long distance to the target parts. The axon membrane is wrapped with a myelin sheath for fastening nerve impulse transmission. The axon membrane is wrapped with a thick myelin sheath for protection against damage. The myelination is interrupted at regular intervals by the node of Ravian, increasing the speed of impulse transmission through salutatory conduction. Part B. With appropriate examples, describe the various types of reflex arcs. Monosynaptic reflex arc. A reflex arc consists of only a single chemical synapse between one sensory neuron and one motor neuron. Example, we have the patella reflex Achilles reflex, muscle spindle reflex. Polysynaptic reflex arc. This is a reflex arc comprising of one or more interneurons connecting the afferent sensory and efferent motor neuron. Compare the neurocells with other cells found in the mammalian body. Part C, we shall first look at the differences. Neurons have specialized extensions called dendrites and auxins. Other cells of the body, they lack the dendrites and auxins. Neurons communicate with each other through electrical-chemical process. Other cells in the body, no such electrochemical communication. Neurons contain specialized structures, for example, the synapse, and chemicals, for example, the neurotransmitters. Other cells of the body, no neurotransmitters. What is the main difference between a generator potential and an action potential? Part B. Discuss the exteroception using suitable examples, highlighting the receptor, stimuli, location, structure, and the process of transduction. Part A. What is the main difference between a generator potential and the action potential? The amplitude of a generator potential is proportional to the stimulus intensity, whereas the amplitude of the action potential is always the same. It obeys the all or nothing law. Discuss exteroception using suitable examples, highlighting receptor, stimuli, location, structure, and the process of transduction. Stimuli, we have gravity. Interoceptors, they are the statocysts. Location, the outer chamber of the inner ear. Structure, there is the otolith and the air cells. Transduction, otolith deform hair cells. Another exteroception is motion as the stimuli. Interoceptors, the chupla, lateral line organ. Location in semicircular canals of the inner ear. Also within the groove of the body surface of the fish. The structure, collection of hair cells, and transduction See, the fluid movement deforms the hair cells. Another exteroception. Stimuli is test. Exteroceptors, test bud cells. Location, mouth, skin of the fish. The structure, they are chemoreceptors, epithelial cells with microvilli. Transduction, chemicals bind to the membrane receptors. Another exteroception is the smell as the stimuli. Exteroceptor, olfactory neurons. Location, nasal passage. The structure, those are chemoreceptors and ciliated neurons. Transduction, chemicals bind to the membrane receptors. Another exteroception stimuli is hearing. Exteroceptors, organ of Corti. Location, cochlea of the inner ear. Structure, hair cells between the basilar and tectorial membrane. Transduction, sound waves in fluid deform membranes. Another exteroception stimuli is vision. Exteroceptor, we have the rods and the cone cells. Location, retina of the eye. The structure, ray of photosensitive pigments. Transduction, light initiate the process that closes ion channels. Another interoception stimuli is heat. Interoceptors, pit organ. Location, face of the snake. Structure, temperature, receptors in two chambers. Transduction, receptor compare temperatures on the surface and the interior chambers. Interoception stimuli, electricity. Interoceptor, ampullae of Lorenzin. Location, within the skin of fish. Structure, closed vesicles with symmetrical ion channel distribution. Transduction, electrical field alters ion distribution on the membranes. Number 45. What is meant by the following term as far as hormone functioning is concerned? 1. Target site. 2. Cascade effect. B. Outline the physiological effect of the catecholamine such as adrenaline. Part C. Distinguish between the resting potential and the generator potential. What is meant by the following terms as far as hormone functioning is concerned? target site. The target site is an organ, tissue, or organ bearing specific receptors for a particular hormone and responds specifically to that hormone. For example, target organ for the action of glucagon in the liver. Cascade effect. Cascade effect refers to a series of steps or stages in a physiological or biochemical process at once initiated by hormones, continues to final steps by virtue of each step being triggered by the preceding one, sometimes with the cumulative effect. Part B. Outline the physiological effect of the catecholamine such as adrenaline. Dilates pupil of the eye. It permits the adequate entry of light in the eye in the times of fear and flight. It causes hair to stand on end. Relaxes bronchioles, thus increasing airflow to the lungs. Inhibits peristalsis and digestion. Prevents bladder contraction. Increase force and the rate of heartbeat. Causes generalized vasoconstriction. Increase blood pressure. Stimulate conversion of liver glycogen to glucose. Decrease sensory threshold. Increases cerebral blood flow, raising mental awareness. Part C. Distinguish between resting potential and generator potential. Resting potential. Attained when the outside of the membrane is more positive relative to the inside. Generator potential. Attained when the inside of the receptor membrane is more positive relative to the outside. Resting potential, negative potential difference. Generator potential, positive potential difference. Resting potential remains constant and never reaches threshold. Generator potential changes and increases to threshold. Resting potential. Sodium ions actively pumped out. Generator potential. Sodium ions rapidly diffuse inside. Thank you very much for participating in coordination and control. Questions 41 to 45. Please do like, share, and subscribe.